speakers. Our first speaker is Professor Sumit Chanda, who's Director and Professor of the Immunity and Pathogenesis Program at Sanford Burnham, previous Medical Discovery Institute in San Diego, California. His lab has been one of the leaders, pioneering lab in studying virus-host interaction through genome-wide screening methodologies and protein-protein interaction analysis. They have varied interests in different kind of viral pathogens. They've been working with influenza, with HIV for a long time, but when the COVID-19 emerged, they were the first one to conduct probably the largest repurposed drug screening ever done in shortest ever time possible. That work was, the scientific part of it was published in Nature. The human story of it was covered in New York Times. So we are really excited to hear about that and maybe much more. He has a lot of new things to discuss. With that, I will invite Professor Chanda to turn on his video and audio. Hi, Sumit. Thank you for joining.
Thank you for the invitation.
Evening in San Diego, and thank you for giving away your family time, dinner time for us to give this talk.
Oh, it's my pleasure. Can you see the screen and can you hear me okay?
Yes.
Yes, okay.
Keep the screen live. Just hold on.
I'm going to shut down my video just because that will help with the bandwidth.
Can you share your screen again, please?
Yes.
Okay, we see it now. You can put it on full screen.
Okay, it should be on full screen.
Ideally, for better visibility.
I have it on full screen. You can't see it on full screen?
Okay, looks like, yeah, it's fine. Please go ahead.
Okay,
Thanks.
okay. Well, Shashank, thanks for the invitation. It's almost a return invitation. I was out there in Bangalore visiting Shashank and IISc about 3 months before the pandemic broke out, and I hope to return one day very soon. So as Shashank mentioned, our lab is a virology lab. We focus on host pathogen interactions, and we, like many other labs, have pivoted to studying COVID. And I'd like to tell you a little bit of a story of how we ended up getting there and what we're doing in the drug repurposing space. So as most of you know, the SARS-CoV-2, the causative agent of COVID-19, most likely originated in a bat and then was subsequently transmitted to humans, most likely through an intermediate species, which the pangolin is thought to be the most likely culprit there. What we don't know is if the virus came preloaded with the instructions for human-to-human transmission, or if this is something that occurred in the population in China, where the virus acquired mutations that enabled a greater spread of the virus. One thing to note that we've been observing through a number of our studies is that there is significant amount of transmission from human to various domesticated pets, and these pets can harbor the virus and replicate the virus in high titers, including hamsters, cats, and even in New York City, there was a report of a tiger in the Bronx Zoo coming down with COVID from one of their trainers. And so this, even with a vaccine, these reservoirs really are something that we need to contend with for domesticated animals. It's unclear what the reverse transmission is between these, but really something to keep an eye on. And the viruses are mutating in the animals. There was a recent report out of Denmark that minks are now being culled because of mutations in the spike domain protein, which is the target for almost all of the vaccines. So this is just a chart of the major respiratory viruses that we've been afflicted with over the last 2 decades. I won't go through the entire slide, but some very important numbers for COVID-19 in comparison to some of the other respiratory viruses like flu is the R0 number. So the R0 number is the number of people an infected patient transmits to. It's much higher for SARS-CoV-2 than it is for flu. The other important data point here is the incubation time for SARS-CoV-2 is highly extended and encompasses a pretty large asymptomatic period where people can spread the virus before knowing that they're infected. So these case fatality rates need to be updated. So they're going down now that we know how to treat the virus, both with drugs and with palliative care. But still, we're about a log order of magnitude away from influenza, which is a manageable disease, even though in the US, influenza still continues to claim 35,000 to 50,000 lives per year. This is just a graph that shows you the impact of the last global pandemic. This is life expectancy in the US, and you can see that the 1918 flu pandemic had a much more profound impact on life expectancy here in the US, even in comparison to the 2 world wars. And the 1918 pandemic, of note, came in multiple waves. So we're undoubtedly now in a global second wave of the SARS-CoV-2 pandemic, which happens as the Northern Hemisphere transitions to the fall season, similarly to the 1918 pandemic. And in 1918, 1919, there was another wave in late winter, early spring, and there's no reason to believe that that won't happen with SARS-CoV-2. One thing to also note is that influenza pandemics don't go away. Those viruses don't go away. They become seasonal. They become endemic. So in 1918, there was the pandemic that claimed 500,000 lives in the US and millions worldwide. But that virus didn't disappear. The H1N1 virus then became seasonal and continued to circulate. And this was not necessarily because of a vaccine, because vaccines to influenza weren't discovered until the 1930s and rolled out in any large scale for some time after that. This was just the natural progression of a pandemic virus. And what happens with influenza is you get reassortment, and then you get sporadic novel pandemics. So in 1957, you had the H2N2 become the dominant strain that led to a pandemic that claimed 70,000 lives in the US. And then 1968, the H3N2, which originated out of Hong Kong, claimed 35,000 lives. And the H3N2 is currently one of the circulating influenza strains that is right now in existence. For unknown reasons, the H2N2 was also replaced by H1N1. There was feared to be in 1977 another potential pandemic, but that didn't materialize, and it's thought because of pre-existing immunity to the H1N1 derived from the 1918 strain, that that novel virus did not produce a significant pandemic. And then, as most of you can remember, in 2009, that H1N1 strain was replaced by the swine origin influenza virus, which thankfully did not trigger a global pandemic that we were all afraid could happen. But this just really illustrates that these respiratory pandemic viruses never go away. They stay, they become seasonal, they become endemic. There are very few viruses that we've ever eradicated, even with wonderful vaccines. And so COVID-19 and SARS-CoV-2, I believe, will be with us for some time to come. And we will just need to learn how to deal with it between vaccines and therapeutics. Oops, sorry. So how did we get into this? So a couple of months after I had traveled back from my trip to India, it was around Christmas time, and I have a postdoc in the lab, incredibly talented postdoc who took a well-deserved holiday back to China to visit his family, and he sent me an email talking about in mid to late December, talking about a cluster of pneumonia patients with unknown etiology in the Hubei province and in Wuhan. And I said, this is interesting, keep me posted. And then he wrote back in late December, early January, that the causative agent was a novel coronavirus. And so for most of you who work in the virus space, when you hear the word novel coronavirus or novel influenza virus, you immediately become incredibly alarmed. So these are viruses with high pandemic potential because they're transmitted through respiratory droplets. So unlike previous threats like Ebola, which is transmitted through close bodily contact, or Zika, which is transmitted through mosquito, this is something that really could readily be transmitted by somebody getting on an airplane and then infecting another continent. So at that time, we decided that we needed to start putting together a team of scientists, the team that I like to call the scientific dream team for coronavirus. So we reached out to Ren Sun, a collaborator at University of California, Los Angeles. At the time, the virus was only available in China. And so we had to collaborate. We reached out and collaborate with Dr. K.Y. Yun and Shofang Yan at Hong Kong University. So Dr. Yun was actually one of the people who identified the SARS, the original SARS in 2002, 2003, and they had the virus cultured and going in the lab, and could immediately start doing experiments. And then Adolfo Garcia Sastre, who used to where Shashank was in Mount Sinai in New York. We also thought to ourselves, what could we do that would be the quickest impact for this virus, which was at that time only confined to China, but we had great trepidation and fear that this is something that could rapidly spread outside and throughout the world. For those of you who are aficionados of drug discovery, you know that it takes somewhere around 10 to 15 years to develop a novel therapeutic or a novel antiviral. And of course, for a pandemic, this is not the kind of timescale that you want to respond to. So we decided to pivot to a repurposing strategy. So a repurposing strategy is essentially taking known drugs that have been in people, so we know that they're safe. They may have or may not have worked for the indication that they were originally developed for, but to see essentially by serendipity whether they might have antiviral efficacy. So of course, remdesivir is a fantastic example of this. Remdesivir was originally developed for hepatitis C, and then Ebola, and now has been approved for the treatment of SARS-CoV-2. So we were fortunate enough to link up with Arnab Chatterjee at the Scripps Research Institute right across the street here in La Jolla. They had been funded by the Bill and Melinda Gates Foundation to build what I believe is the most extensive repurposing library containing 12,000 known drugs. And the plan was that a postdoc in my lab, Laura Riva, would fly to Hong Kong and work with Dr. Yoon's group to show them how to screen 12,000 drugs. So for those of you who are not virologists, typically labs can screen maybe 2, 3, 5 drugs at a time, but it really takes a special skill set that Laura had to be able to look at 12,000 different compounds for antiviral activity in rapid succession. So just as she was going to pack up her suitcase with a library, some equipment and automation from our lab, travel to China was shut down by the US. She could actually leave, but she wouldn't be able to come back. And so we had to come up with another plan on how we could transfer our knowledge and expertise of screening these kinds of large-scale libraries to Dr. Yoon's group, who had been culturing the virus and had it replicating. And so what we settled on was doing high-throughput screening by iPhone. And so we downloaded an app called WhatsApp, and then we also shipped all of our equipment and libraries and reagents out to Hong Kong. Of course, shipping during a pandemic is never easy. The package ended up in Guangzhou, not Hong Kong. Some of the materials were on dry ice, so of course we started to panic. My lab manager, Paul De Jesus, was on the phone 24/7 with Federal Express, making sure everything was refilled. And fortunately for us, everything got there filled with dry ice. And you can see this is some of our robotics that are in a BSL-3 hood in Dr. Yoon's lab in Hong Kong. And because WhatsApp is not available in China, we downloaded an app called WeChat. And Laura would get up in the middle of the night, and they had a dedicated iPhone for their BSL-3 facility, and would video conference and instruct the folks in the Hong Kong lab how to do this kind of high throughput screening. And as Shashank mentioned, their incredible efforts were chronicled in the New York Times. I really am not doing it justice. So if anybody's interested in reading that, there's a nice article there. So getting back to the science, how was this screen set up? So we used ViroCells. So again, for you virologists in the audience, you know, ViroCells are a favorite of virologists because they lack interferon signaling. And so they tend to support viral replication pretty well. So that is the cell system that they had going in Hong Kong. And the other lucky break that we had was that this virus, when you infect these ViroCells, cause CPE or cytopathic effect, which means essentially they end up killing the cells. So you could use cell death as a surrogate for measuring viral replication. So the quantification of the cell death is over here on the right-hand side. It's about a 1.5-fold window. It wasn't great, but in the middle of an emerging pandemic, we couldn't wait for a perfect. So again, we also shipped out this reframe library, which is the repurposing library. You can see what the breakdown is. 39% of them are approved drugs. 58% of them are investigational new drugs. So this means that they have safety data and pharmacological data and tox data in people. They've been in phase 1, but hadn't progressed further for a number of different reasons. A couple of unique things about this library: almost 80% of them are not found in other repurposing library, and 40% of them are not found commercially at all and required de novo synthesis. So the flow of the assay was that these compounds were spotted in 384 well plates, 1 compound per well. Then we added Vero E6 cells, challenged with the virus, and then measured cell death. And anything that could rescue the cell death we considered to be potential antivirals. This is just some of the data showing the reproducibility of the assay. So we did a primary screen and duplicate, and then the actual screen, and we had reasonable separation. This is not something that we probably would have done. We would have kept optimizing this if we weren't under these situations, but we had to go with what we had to go with. We had a nice positive control. At the time, remdesivir was not identified to be a real potent antiviral, so we used basically an endosomal inhibitor as our positive control. But you can see, for the most part, we got good agreement between the 2 replicates of the screen. There were some outliers, but we really decided that we wanted to power through this. So these represent the hits in the screen that rescued the cytopathic effect of the virus. And so one of the things that you can do with this, because these are known drugs with known targets, you can look for target classes that are overrepresented in your screen. And so what I'm showing here is a number of different target classes that were overrepresented. You can see the anti-malarial class, hydroxychloroquine and quinolones. These have always worked great in cell culture, never in vivo or in animal models. So this wasn't a terrible surprise to us that these were highly enriched. Another class was retinoic acid receptors. So let me see if I can go back here. So I can just show you what this analysis is. Each dash represents a different molecule in that class of drugs, and higher activity of those drugs are to the left. And so the more enrichment you have of this class of molecules to the left, the more confident that you can be that this mechanism of action is really truly antiviral. And you could also do some other tricks where, in this case, we had retinoic acid receptor agonists, and we could go in with antagonists to see if we can reverse the phenotype. And in this case, you could. Here's what the agonist does, and then you add the antagonist, and you see a reversal of the phenotype. Okay. So during this time, we started getting access to the virus here in the U.S. So Laura Martin Sancho, who's another incredible postdoc in the lab, was the first person in San Diego to receive the virus, and really heroically, within a week, had it up and going at titers that we can use to do confirmatory screens. One of the confirmatory screens that she developed was this immunostaining screen. It was an antibody provided by Adolfo Garcia Sastre that he had raised against the N protein of the original SARS virus and put it away in a freezer and then thawed it out. And about 15 years later, we found that it had really great cross-reactivity to SARS-CoV-2, and we could now directly track replication of the virus instead of using this surrogate readout for cytopathic effect. So we use this assay, and originally we had selected 300 compounds from our screen, and using this assay, we were able to validate 100 of these compounds to work to cause greater than 40% inhibition of SARS-CoV-2. And this is just a cartoon of where these compounds are thought to work in the cell. You can see that other than some of these factors that work at the endosome or work at the ER or the Golgi, most don't directly intersect with the viral life cycle. So we think that this suggests that these are not direct antiviral effectors, but they are targeting host molecules that the virus requires for replication, and these are regulators of those molecules that promote viral replication within these cells. And so you can see GPCRs, ion channels, nuclear transcription factors. The virus never actually makes it into the nucleus. So it's a reasonable hypothesis that very few, other than remdesivir, which was pulled out on the screen, were directly acting on the virus. So then what we wanted to do was look to see how many of these actually confirmed in dose response. So 2.5 micromolar is a pretty high concentration and not something that is readily achievable clinically in patients. For most compounds, not all, but most compounds, you typically need triple digit to double digit to triple digit nanomolar cellular EC50s. And so we zeroed in on about 21 compounds that had EC50s. So these are kind of the more, the left is a heat map, the right is a more traditional EC50 plot of these drugs. There were several classes that popped up. I mentioned GPCRs, chloroquine derivatives, ion channels. There were a couple of antivirals that I'll talk to you about. Remdesivir at that time, remdesivir had already begun clinical trials, and some cysteine proteases, and we did some more investigation of those. The other thing I want to point out to you is the measurement of what we call a selectivity index. So we like to be at or near 1 micromolar EC50, so 50% inhibition at that concentration. But the other thing that's important is selectivity index. So the difference between the cytotoxicity of the drug, which is depicted in red, and the antiviral efficacy. We found numerous times that if you make cells sick, they can't do anything well, including support viral replication. So a lot of times you get false positives when you start chasing down cytostatic compounds or cytopathic compounds. So I'm going to tell you a little bit about some of the molecules that we find to be most interesting, and that we built a data package around. So this one was interesting to us because it was an HIV reverse transcriptase inhibitor. Reverse transcriptase is the HIV encoded polymerase, and so this and it's a non-nucleotide reverse transcriptase. So this suggested that this might be an inhibitor, another inhibitor of the coronavirus polymerase, and perhaps an allosteric inhibitor, and can give you similar activities as remdesivir, but targeting a different pocket of the molecule. And so we're running viral polymerase assays right now to really nail this down. Its EC50s are commensurate to what's observed with HIV, and we're now working with Janssen, who owns this molecule, to get enough to test it in animal models. So, of course, I mentioned we did all of our work in Vero E6 cells. So Vero E6 cells are a non-human primate kidney epithelial cell. And so one of the concerns was that these might be cell type specific drugs, and not necessarily things that can work into humans or across different cell lines. So we tried these 21 drugs in 2 different human cell lines. So 293, which is another kidney epithelial cell, and H2H7, which is a liver-derived cell, both transfected with ACE2. 18 out of 21 of those drugs had activity in 1 or more of these cell lines, suggesting that a majority of these molecules were not either monkey specific or Vero E6 specific. The next thing we did was to zero down on a few of these molecules, do additional characterization. One of the things we wanted to look at, because at that time remdesivir was looking pretty promising in the clinic. The problem with remdesivir, as most of you know, it's a very hard molecule to synthesize. It's a 21-step synthesis and extremely expensive, and there's a limited supply in the world. And so we wanted to see if any of our drugs had any sort of synergy with remdesivir. And so what I'm showing you here is a graph of each drug given the EC50 without remdesivir. The predicted EC50 is the red dotted line when you combine remdesivir, and the observed EC50 is the red solid line. And that red shading area really represents the deviation from expected to observed. And each one of these four drugs had some level of synergy with remdesivir. And I'll talk to you a little bit about what each one of these drugs are coming up. So the two drugs I want to focus on that had this synergy, the one, it's called Henfenshing A. It's a phase 3 clinical compound that's been in the clinics for malaria and cancer. It's been previously shown to block Ebola entry through blocking endosomal calcium channels, and of course this is a member of the hydroxyquinoline family. There are some issues with toxicity, but we are looking at molecules that can be given at low enough levels where you don't have potential cardiotox, but still can enhance remdesivir activity. Another molecule is a phase 1 clinical compound. It's a mood disorder compound targeting the HT1B receptor antagonist receptor. It's an antagonist of that receptor. We really don't know or have a good hypothesis on how this might be affecting replication, but nonetheless, this is a molecule that we've not only found inhibits SARS-CoV-2, but synergizes with remdesivir. So I want to drill down some more and focus on some of the most potent compounds that we pulled out. A huge class of protease inhibitors and one kinase inhibitor. And I want to tell you a little bit about what we learned about these molecules. And so we decided to look at their antiviral mechanism of action by trying to stage where in the viral life cycle they're working. So the first thing we did, what is known as time of addition assay. So typically you pre-treat cells with the drug, add the virus, and then look for inhibition. You can then stagger the treatment, right, where you give the drug either before or after adding the virus. And so if a drug works after adding the virus, it suggests that it impacts a post-entry step. But if the drug only works at the same time or before, it suggests that it targets an entry step. So all of these potent molecules that I mentioned to you, the kinase and the protease inhibitors, only inhibited the virus significantly when you add them early. So that suggests that these are working at viral entry steps. So Xin Yin in the lab developed a pseudotype assay where he took a VSV pseudotype virus and pseudotyped in either a SARS-CoV-2 spike protein, a MERS spike protein, or the VSV-G protein. So now you have entry only dependent on those respective entry mediators for those viruses. And what you can see here is that all of these compounds were blocking spike-dependent, SARS-CoV-2 spike-dependent entry, really convincingly showing us that they are blocking viral entry. So our first hypothesis was that these are mostly protease inhibitors. Maybe they are blocking SARS proteases. SARS encodes 2 proteases, and some of these, based on structures of both the proteases and the small molecules, were really thought to impact that. But Andrew Messakar at Purdue University ran biochemical assays against these proteases, and they were inactive in those assays. So we concluded that these molecules were targeting host-directed proteases that are likely endosomal proteases that are required for SARS-CoV-2 cleavage. And I'll tell you spike cleavage, and I'll tell you a little bit about some of these molecules and what they do. So ZLVG was a molecule that we found that was incredibly potent. This was as potent as remdesivir, almost as potent as remdesivir. It was developed as a cysteine protease to target a cysteine protease produced by group A strep, but we believe that the molecule is functioning by targeting endosomal proteases required for spike maturation. Similarly, VBY is a phase 0 compound that targets endosomal cathepsins, and cathepsins also have been implicated in that entry step of SARS-CoV-2. VBY was one of the molecules that was synergistic with remdesivir, and we believe that it is working through blocking endosomal processing of spike. So the next thing we did was move away from these cell systems, these cell line systems that we were working, and you have to remember, we were really making this up as we went along. No one knew what cell systems this virus could replicate in. So fortunately, we had a number of fantastic collaborators, including our Adolfo Garcia Sastre, who had set up a system where he derived pneumocytes, which are the lung cells that are the predominant site of replication of SARS-CoV-2. So these were derived from iPS cells, and so he was able to show robust replication. And when you treat these cells with remdesivir, you can knock viral replication down. Also, with 3 of the drugs that we tested, ONO, MDL, and epilimod, and I'll tell you a little bit what those are. So ONO, again, is a cathepsin inhibitor like BBY. We think that it's working, again, through blocking cathepsin activity in the endosome and blocking the processing and maturation of spike that allows the escape of the virus from the endosome. We're now working with Ono Pharmaceuticals to obtain this drug and move some of these compounds into in vivo models. MDL is a late preclinical compound, again, a protease inhibitor. It's originally developed as a non-lysosomal protease inhibitor, but we have every reason to believe that its antiviral activity is working through disruption of endosomal proteases. The next assay that we were able to exploit was a lung explant assay. So this is essentially taking biopsy lung tissue from a patient in Hong Kong, culturing them in the lab, and then challenging them with virus. And the molecule that we tried was a molecule called epilimod, which is now in phase 2 clinical trials for SARS-CoV-2 across the US. And as you can see here, we find that epilimod works as well, if not better than remdesivir in this specific primary cell system. So what is remdesivir? It's a phase 2 compound. It's a PIK5 kinase inhibitor. PIK5 kinase inhibits endocytosis. So the virus uses endocytosis to get into the cell and then into the cytoplasm. And we believe, based on our data, that epilimod is blocking that essential step in viral replication. It is incredibly potent, most potent molecule that we've seen, and has activity against Ebola virus also. And I would expect it to have activity against any virus that uses this method to enter the cell. So our next step is to move things into in vivo models, and I'll talk to you about one of the molecules that we've progressed the furthest, and I haven't talked about this as of yet, but the reason that we progressed this was not because it was the best molecule that came through the screen, but for reasons that are more specific to the time and place that we are. This molecule called clofazimine or lamprene is a leprosy Hansen patient drug. It's an antimicrobacterial. It has been around for the last 40 years, so we know it's incredibly safe. It is orally bioavailable, so you can give it to patients on an outpatient basis, whereas remdesivir requires an IV push, and it's cheap. It's about $1 a day in the US. It's even cheaper in India compared to a $3,000 treatment course of remdesivir. It had a pretty good efficacy. Again, it wasn't a pilimod, but it did do well enough where we wanted to move that further. And so we wanted to see how it was working. And so similar to the other drugs I showed you, it also could block in our entry assay. But the other surprising thing was it also blocked synthesis of viral RNA. So what we did was took viral RNA, transfected it in. So SARS-CoV-2 is a positive strand virus. We transfected it in, so you would bypass the entry step, and then look for negative strand genome. So these subgenomic negative strand fragments, and we could see that clofazimine both blocked entry and the synthesis step. We're currently investigating how they're both working in both steps. And we believe that there might be some inhibition of viral helicase activity, but we're still working to figure that out. But what got us really excited about this was the activity of clofazimine in the hamster model. So the hamster model is probably the best small animal model that recapitulates human disease. The hamster gets inflammation and immune response after SARS-CoV-2 challenge. So that's why we chose the hamster model. This work was done by Shofang Yang in University of Hong Kong. And you can see in the prophylactic setting, we were able to knock down lung titers by about a log and a half. Almost as interesting was that you can really see a behavioral change in these animals, whereas these untreated animals are very lethargic. They tend to be hunched over. The clofazimine treated animals were walking around and behaving as normal. The therapeutic administration wasn't as dramatic, a little under a log, but reasonably comparable to remdesivir. Clofazimine is in red here, and remdesivir is in blue. The lung histology in prophylaxis setting looks like mock infected lung. The therapeutic setting, there's still some inflammation in there, but reasonably comparable to remdesivir. And if you look at the immune response to the virus, looking at IL-6, TNF-alpha, and CCR4, these are all cytokines that are induced by the virus, you can see that we see a pretty dramatic suppression of the inflammatory response to the virus. The other exciting thing was that clofazimine was found to synergize with remdesivir. I won't go through all of the data here, but the take-home messages on the left-hand side, the more red, the more synergy, and the right-hand side are similar to the graphs I showed you. The black is remdesivir alone. The dotted lines are what you would expect if they were just additive, and the observed are the blue and the red solid lines. But essentially, the take-home message here is that if you give clofazimine at therapeutically achievable doses, you can use about 1/20th of the amount of remdesivir required for antiviral efficacy. So before I finish up, I want to share with you a quick anecdote and then appeal to you for some help. So I mentioned that clofazimine is a frontline treatment for people with Hansen disease or leprosy. And we almost serendipitously started conversing with a clinician in Brazil who was treating Hansen's patients and was writing up a review on the noticeably low incidence of COVID-19 in these patients. And she hadn't seen our data on clofazimine. And so when she went back and she looked, and she found that of 150 patients that were on clofazimine, none came down with COVID, and the 52 that were taking some other drug, 3 had COVID. So these are not statistically significant data, but we believe that the clinical trial for clofazimine, at least in a prophylactic setting, has been done. So if you or anyone that you work with has access to clinical data on COVID incidence in Hansen's disease leprosy patients, plus minus clofazimine, please reach out to me. We would love to work with you to get this data, to amalgamate this data, to see if clofazimine can be really have some level of prophylactic benefit that the small data set that we're looking at really seems to indicate that it does, in conjunction with both our animal model study and our cell study. So what is this path forward? Remdesivir is the standard of care right now. There are a number of different other therapeutics that are on the horizon. Eli Lilly just had an antibody approved that are neutralizing antibodies, convalescent plasma, a very promising molecule, MK44A2, which is a polymerase inhibitor, looks like it will do well in the clinics. Interferon. And then there's also the immune suppressors. I think this is going to be key. Controlling the virus is not going to be enough. I think controlling the virus is necessary, but not sufficient. So suppressing cytokine storm, either with dexamethasone, cytokine inhibitors, or JAK inhibitors like baricitinib, are going to be critically important in a therapeutic regimen. And then we're also looking at things like efficacy, cost and availability, delivery. Can you give it to patients in an outpatient setting? Can you give it safely to patients? Can you give it in a prophylaxis setting? Is it safe enough to give in a prophylaxis setting? And finally, resistance. We're not thinking much about resistance now, but we should be thinking long term. The best way to combat resistance is in cocktails. We know this with HIV, we know this with HCV, and we're going to have to find a combo resistance, a combo antiviral cocktail that can circumvent resistance also for SARS-CoV-2. So I want to really thank you for your attention. Also, I thank the people who we collaborated with along the way, and also the members of my lab for really for last 9 months, heroic efforts, really working day and night to try to get what I would consider 4 or 5 years worth of work done in such a short amount of time. So thank you very much.
Thanks a lot, Sumit. You can stop sharing your screen, and then we'll take up a few quick questions.
Great.
Okay, so first question is from Professor Kumar, Chair of MCB department here. He's asking, would it be a good idea to have a combination of antiviral targeting a viral factor plus antiviral targeting a host factor, say Epilimod and remdesivir?
So I think that's that there are some other antivirals being developed against protease and some other things, but host factors are nice because it's much harder for a virus to develop resistance against them, because the small molecule is not directly binding to a pocket in the virus where a single amino acid change can render a drug useless. What a host factor, resistance to a host factor, really requires a rewiring of the host pathogen interactions. So we really think that a combination of host and viral antivirals are going to be key in developing these cocktails.
Yeah, second kind of a question and a comment as well from Amit Singh, my colleague here at CIDR. He's asking that whether or not you will expect hydroxychloroquine to have synergy with protease inhibitor, which also required pH stop in endosome. And
yeah. So hydroxychloroquine is an interesting question. So I mean, this is Shashank, you know this, right? This was also it works against flu in vitro, really great. Never worked in vivo, and so it's unclear why. And so my favorite hypothesis is that you just can't get enough concentration. So in vivo and to achieve therapeutic efficacy. So where we're seeing synergies is actually when you attack two different parts of the viral life cycle and not the same. So assuming that the preponderance of the protease inhibitors will be working at a viral entry, and you throwing another viral entry inhibitor probably won't get you synergy, but I wouldn't be surprised if, say, remdesivir or something else would be synergistic.
Amit also suggests one point that clofazimine, it might have RNA poly inhibition activity because it sequesters metal ions which are required for the activity of protocol. I have one question, and after that another one, and then we'll end the Q&A. Do you plan to repeat your screen on, say, neuronal cell or endothelial cell? Because few things may work in one cell types and you may have missed out. Virus, now we know it goes to other cell types also.
Yeah, so not neuronal cells, but we're looking to repeat this in primary epithelial cells. So these iPS-derived epithelial cells, and we're also going to do these synergy screens. So we're going to add suboptimal levels of remdesivir into primary cells and then repeat them. So if we found things that either only work in epithelial primary cells but not cell lines, and may synergize with remdesivir but not have real profound monotherapeutic activity. So that is ongoing. We're working with a couple of groups who are much better at growing these primary cells than we are, and scaling them. That's the hard part, is scaling them.
Okay, last question, not really related to drug discovery, but what are your thoughts on really low case fatalities seen in India? In India, it's close to 0.1%, which is similar to flu actually. I mean, numbers are high in general because the population is large, but statistically speaking, it's similar to flu.
So there are two mysteries, right, of COVID that I'm trying to understand. So one is in Africa, in sub-Saharan Africa, right? We expected that because of their healthcare infrastructure and the preponderance of African Americans that got the disease, that sub-Saharan Africa would actually feel a huge brunt of this. And the second is India, right? So the low case fatality rate in India. So the hypothesis is, or could be, that because in the U.S. we're so germophobic that we don't get encountered to a lot of different pathogens, so our immune systems have not built a repertoire, and so we know that other coronavirus infections can protect you from SARS-CoV-2, especially with T-cell immunity, right? And it could be that protection has been affording people in India to have less case fatality rate. It could be, there could be a genetic basis to it. That hasn't been ruled out. But it also could be environmental. Big mysteries, but those are the two hypotheses: the genetic hypotheses and the fact that the U.S. are really germaphobes here and really go overboard in cleaning and sanitizing, which doesn't allow our children's immune system to develop in ways that can be favorable as you become an adult.
Yeah, more reasons to visit India often. All right.
Agreed.
Thank you so much for joining. I'll ask you to